Hello everybody. If you're one of the students who didn't get their exams back for one or the other reason in the labs, then they're in my possession. I have them right here. You can pick them up after the class in that case. The zip strips, you can see those on the website. So that tells you what you did or didn't get correct on the multiple choice part. And you have to go to the folder called course content, and then there's a link, and then you have to log in from there to find your results. Alrighty, today we will we'll finish our look at membrane electrochemical gradients, and then we'll start into metabolism and respiration. So we've seen this system. If we produce a vesicle enclosed by a membrane with a high potassium inside and low potassium outside, 10-fold concentration ratio here, then if we add cholinomycin, we have to remember that cholinomycin is an ionophore, which you should know, and it's an ionophore that carries potassium. Similarly, you should remember that nigericin exchanges potassium for protons. So if we add cholinomycin, we have a pathway for potassium. Potassium flows out, leaving a negatively charged chlorine behind. We get a membrane potential, and the Nernst equation tells us that there will be 60 millivolts size magnitude of the membrane potential. But we can immediately see that it will be 60 millivolts because a 10-fold concentration ratio for potassium, which drives the formation of the membrane potential. That's one standard package. So we get a one standard package size membrane potential, 60 millivolts. Now we can ask the question, can we actually measure that membrane potential in some kind of a way? And we can do that by adding another molecule, which is not an ionophore, but it is an ion itself, and it can move through the membrane. It's lipophilic, it can move through the membrane. It's positively charged, so this tetraphenyl phosphonium, TPP+. If we add a low amount on the outside, the significance of the low amount is that it will not overcome the potassium concentration gradient. So we add a low amount, and this stuff can be labeled using carbon-14. So we add this from the outside, and we have a membrane potential that's positive out, negative in, and the TPP+ will flow in. And it will distribute itself according to the Nernst equation, which means we're getting with this 60 millivolts membrane potential, or one standard package, we're getting a 10-fold concentration ratio. The TPP accumulates, so it's higher inside, so we get approximately 10 nanomolar TPP+ inside. Okay, now this is actually something that can have an application. You know we can measure membrane potentials in large cells by directly sticking electrodes in. But if you want to measure the membrane potential over an organelle membrane, maybe the mitochondria, or if you have bacteria which are small, then you cannot do anything with your electrodes. They're just too big. The cells are too small, the compartments are too small. So you can use the distribution of labeled ionic substances such as TPP+. So let's imagine we have here a suspension of small, let's say, bacteria. And these are alive, and they have a membrane potential over their plasma membrane. Can we measure that membrane potential? Yes, we can. If we add the TPP+ to the outside, then if the membrane potential is as expected, positive out, negative in, the TPP will accumulate into these cells to a concentration that is predicted by the Nernst equation. So can we measure that? Yes, we can. Yes, we can. So I have Use a little bit of oil, and this is a special oil, mineral oil, based on silicone, I think. I'm not sure to be sure exactly what type of oil that is, chemically. But the important point is the oil is heavier than water, but lighter than the cells. So if we centrifuge this tube, the cells move through the oil and get perfectly separated from the surrounding medium. Then we freeze this tube, and then you find a volunteer, or you do it yourself, you find a volunteer to actually cut this radiolabeled tube, and you cut it right here, right underneath the surface of the oil, so you separate the fluid above and the cells below. So really what you do is you freeze the tube and then you cut it. So now we can count, determine the amount of TPP plus above in the medium and below, that's in the cells. The TPP plus has 24 carbons, so we can label it with carbon 14. But we don't need the amount of TPP, we need the concentration. And concentration by definition is amount in a volume. To get the volume, we need to label the water as well. So we are not only adding the carbon 14 labeled TPP, we're also adding a little bit of water that is labeled with another isotope, tritium. And these isotopes, tritium and carbon 14, can be counted very accurately, very precisely. So, the amount of tritium label in the fluid above gives us the volume that originally was outside of the cells, and the carbon 14 gives us the TPP plus outside the cells. So we get a concentration outside, and below the oil we get a volume for water from the tritium counts, the amount of TPP from the carbon counts, and therefore from the ratio, the concentration of TPP. So from concentration ratios we can calculate the membrane potential. So in this example, if above we have 1,000 counts per minute, that's the unit that you have for radioactivity for the tritium. So our volume we can say is 1,000. Our carbon amount is 100, so that concentration is 100 over 1,000 is 0.1. And below we have a concentration of 1,000 over 100 is 10. So when you do all the math, you find that you have a 100-fold accumulation of TPP plus below. That means it was in the cells. So that means we have, according to the Nernst equation, 120 millivolt membrane potential. And you can use the Nernst equation or the idea of standard packages. This is, I call this a key diagram because it is a guaranteed exam question. Guaranteed. You know I can always change the numbers for tritium and carbon counts here, above and below. So we can come up with any membrane potential. Your intuition tells you that if these are mitochondria or if these are bacteria, then we're going to get the membrane potential positive out, negative inside the cells. And this is, by the way, what TPP accumulation tells you. If we have another membrane system, maybe if we have isolated thylakoid membranes from a chloroplast, then actually we might get the TPP excluded from that compartment, because there the membrane potential is such that it is positive in the lumen. We'll see that later. So practice with this type of problem. Okay, we're done with this. I've posted task number 3. I think it's active. Some people have tried it. It's not easy, but once you start to look at it more carefully, and I think in the course of next week, you'll find out it's actually pretty doable. And in the end, most students figure it out. The task also gives you feedback if you give some wrong answers, and you have a total of 3 tries. It should work. People have asked how it works with the tasks. Remember, the tasks, there are 8 in total. They're each worth 10 points, so there's 80 possible points. These are fake points, task points. They're worth nothing by itself. But if you get enough task points, 56 over all the 8 tasks, or that's 80% over 7 of them, which everybody can get, then I figure you've done enough to practice and you earn the right to later do the exam corrections. The exam corrections are over midterm 1 and 2, and we'll all deal with that after the spring break. Way after the spring break, we'll deal with that. You get the chance to earn 40 points back. Okie doke. So let's look at metabolism. Yoo-hoo, metabolism. Okay. Metabolism. There's a couple of things you need to know, and I will emphasize those. There are a couple of slides here that I'm not going to go over extensively because it's boring and you can read it. I know you can read it and you can do some things yourself. But here are a couple of things that we need to know. Metabolism is, by definition, all the chemical reactions that occur in an organism. All the chemistry, everything, that's metabolism. And we can divide metabolism in a couple of ways. One division is we can distinguish the build-up reactions and the breakdown reactions. Build-up reactions are called the anabolic pathways. Breakdown reactions are called catabolic. Catabolic. It's all from Greek. Breakdown, anabolic, build-up. You can remember that, anabolic. The bodybuilders, they like to do some. Nobody here, of course, is CSU, especially not on the football team at CSU. Nobody here is using anabolic steroids, of course. The people who do that want that because they want to get big. Build up. So, well, let's look at the anabolic pathways first. They're synthetic, that means you build up things. In contrast, catabolic pathways tend to be degradative. You break things down. You go from larger to smaller. Anabolic pathways tend to overall require energy input. Another word for that is they are endergonic. And catabolic pathways, breakdown pathways, are generally exergonic, release energy, release heat. In general, anabolic pathways are divergent because we go from a couple of small metabolites, essentially acetate, intermediates of the process that we call the Krebs cycle, intermediates of glycolysis and the 20 amino acids, and you go and you build all kinds of different molecules and macromolecules. There's a lot of diversity there, so you go from a simple group of a few small molecules to combine those in bigger molecules that are divergent. So there's different kinds. Okay. In contrast, catabolic pathways are convergent. We go from different molecules that are broken down to ultimately produce the elemental structures, the most important one being acetate. So catabolic pathways converge on acetate, you can say. They're convergent overall. In general, we need to add hydrogen in order to reduce molecules in the anabolic buildup pathways. That's called a reduction. So anabolic pathways are overall reductive, whereas catabolic pathways tend to proceed with the addition of oxygen, so they're oxidative. In general, catabolic pathways produce ATP, and anabolic pathways use ATP. Guess where that ATP comes from? Mainly from the catabolic pathways. So they are linked. There's an intermediate, a coenzyme, which captures the hydrogen that is removed from molecules in the breakdown pathways, and that's the NADH cofactor. NADH is the charged state of that molecule. We'll look at it later. In contrast, in anabolic pathways, we typically have a very similar molecule of NADPH, where there's an extra phosphate. So, enzymes that function in build-up reactions in anabolic pathways have pockets that recognize that extra P on the NADP. Whereas enzymes that function in breakdown reactions have a binding pocket that recognizes just the NAD. So, in general, enzymes involved in breakdown reactions and in build-up reactions are separate entities in the cell. Alright, so we've seen that the ATP used in anabolic pathways typically comes from the catabolic pathways. So the cellular energy and the important intermediate there is ATP, as we've seen before. This is a coenzyme. It's readily available in all cells. We also need to capture hydrogen that's taken from organic molecules, and it's captured in NADH. That's the catabolic coenzyme, whereas for the anabolic reactions we have NADPH. You should remember this distinction, what's in red. In fact, everything that's on there you should know. And it's easy. There's nothing. Okay. It's not a distinction that we can make. We can say there's a metabolism that every cell has, and then there are forms of metabolism, branches of metabolism, that are not found necessarily in every cell or in every organism. So the metabolism that's common to all of your cells, that's, for instance, whatever you need for the synthesis of DNA, RNA, proteins, respiration, and a lot of other pathways, we can call that housekeeping metabolism. Some people really don't like that term, housekeeping, household metabolism. I don't have a huge problem with it. I don't have a problem with it. So that then means that the enzymes that are doing the job for this general metabolism that every cell has. These enzymes are encoded, of course, by genes, and we tend to call those genes housekeeping genes. I'm personally not offended by that term. Some people are. I do vacuum cleaning at home. I'm not offended by the term. Okay, then there's also special metabolism that we do not necessarily find in every cell. There's some specialization. Think in animals, you have cells that store fats. They're specialized in storing fat molecules. So that's not really a type of metabolism you find in that form in every cell. But really, in terms of chemistry, where we do find the special metabolism is in the plants and the microbes. And especially in plants, we speak of secondary metabolism, which is chemistry that involves the formation of special molecules. Many of those are toxins, or they're pigments, dyes to, say, color the flowers of plants. And toxins that just defend the plant, so all the opiates, etc. That some plants produce, or alkaloids, those are toxins, and they just defend the plant, make it less attractive for animals to eat them. But this is specialized metabolism, and each plant is different in this respect. Microbes also tend to have special capacity to break down molecules. You can almost always find a microbe that can break down any weird organic molecule that you can think of. Even if it's chlorinated or fluorinated or something like that. Okay, so there's two distinctions we can make. Alright. So, if we look at metabolism, what is the big picture of life? We'll come back to this later with photosynthesis. But for now, we can take our own view on what's going on the planet in terms of energy. That is just our view. We are heterotrophs. We eat other organisms. And from our perspective, what's going on is there's CO2 and water, and there are photosynthesizing organisms that use that CO2 and water in order to produce our food. That's themselves, the plants. And then that food, which is reduced carbon, is broken down by us. We form CO2 and we form water in that process. So what is actually going on? The photosynthesizers, that's the plants, the algae, they split water, they take electrons from water, and they capture that energy in NADPH, and they also capture some energy in ATP. So the hydrogen from water is captured in NADPH. There's a lot of energy in that molecule, and there's also energy in ATP. This happens under the influence of light. That ATP and NADPH, that energy is used for the reduction, the fixation of CO2 to form sugars, fats, amino acids, structures that plants and algae make, which we eat. And then those organic molecules can be broken down to CO2 and water in the process that we call respiration. In that process, we utilize oxygen to oxidize our organic molecules. The oxygen was produced as a byproduct of photosynthesis. And in this breakdown of food, we temporarily store high energy electrons in NADH, and we also store energy in ATP. So we have high energy electrons in food and in NADPH and NADH. We have low energy electrons in CO2 and in water. Another question is, what are high and low energy electrons? What is that? Let's answer that question. Well, to answer that question, we have to understand redox reactions. And effectively, we can say that every chemical reaction is a reaction that involves the transfer of electrons, and that is what a redox reaction is. So in redox reactions, any chemical reaction, there's transfer of electrons. There are two extremes that we can think of. In the case of ion formation, we can have a metal and here chlorine, and if sodium is allowed to react with chlorine, Cl2 would be, then the chlorine is very electronegative. It has a nucleus that likes to have more electrons to surround it, and it wants to steal an electron from wherever it can. In this case, it can steal it from the sodium metal, creating the sodium ion, and in the process, chlorine becomes an ion itself. So together, these two ions form a salt. If they're not dissolved, then we have ionic bonds between them. So this is an example of a complete transfer of an electron. That's a clear redox reaction. Now, in biology, we are dealing with carbon, and carbon forms covalent bonds. But we still can talk about electron transfer reactions, because in covalent bonds, the electron pairs are not always shared evenly. We can see that if we look at this series. So methane, natural gas, is carbon surrounded by 4 hydrogen. Hydrogen and carbon are close in their electronegativity. So the pool on these electron pairs between hydrogen and carbon is kind of the same. And so we can say carbon is fairly reduced. You can even say carbon wins out over hydrogen. Now if we gradually oxidize this molecule, react with oxygen, we get methanol. And in this electron pair, oxygen, which is very electronegative, pulls this electron pair closer to that oxygen nucleus, away from the carbon. We oxidize more, ultimately we get that CO2. These two oxygens in carbon dioxide strongly pull those electron pairs towards their nucleus, away from carbon. So the electrons are not shared evenly in this type of configuration. So we can actually say carbon has lost its electrons, and we call that an oxidation. Alright. So, in biology we can say that the electrons fall to the more electronegative group, and in the context of biology, oxygen and nitrogen are the most relevant electronegative atoms. Oxygen being the most important. So if an electron pair or an electron is actually falling closer to a nucleus, you can think about a situation similar to an object with mass falling closer to the surface of a planet. There's potential energy there that can be utilized. So there's potential energy in organic molecules which have bonds between carbon-carbon and carbon-hydrogen. And because oxygen can come in and can accept the electrons from the carbon, that sets of electrons can fall to oxygen. That potential energy can be captured. And cells capture that in the coenzymes, especially NADH, sometimes NADP in photosynthesis, and a little bit also in ATP. So if we look from the perspective of carbon, we can say that if we have a carbon-hydrogen or carbon-carbon bond, then the electron pairs there, those are high-energy electrons. But if we're looking at molecules with oxygen in there, carbon-oxygen bonds are low-energy bonds. That's the idea. So as we go from this high-energy to low-energy situation, the trick is that cells can capture the potential energy in the coenzymes here. It's the big trick. So how much energy is associated with redox reactions? This is something that can be measured. We can measure what's called the standard redox potential. Use an electrode setup, and you get numbers. And typically, the numbers are given for half reactions. For instance, let's consider here the reaction where we have here hydrogen that would react with oxygen to form water. So we can talk about two half reactions. First, hydrogen has to give up its electrons, so you get protons formed. And then those protons and the electrons can react with oxygen to form water. Those are the two half reactions. And there are standard potential differences that we have for those two reactions. If we add this up, then you get an overall potential. Now, if we look at an important coenzyme in biology, there we see NADH, NAD. We see that actually the standard redox potential is very close to that for the half reaction where hydrogen is split. And in fact, NADH is a hydrogen donor, and hydrogen is obviously a hydrogen donor. And they both can donate to oxygen ultimately in a set of reactions where water is formed. The point I'd like to make is that if we look at the total energy here that we have for NADH, we have this value, and it comes out to 53 kilocalories per mole if we convert from a potential to kilocalories. I'm thinking 53 calories per mole, big deal. What does that mean to you? I think I can convince you that the numbers for hydrogen and NADH going to water are going to be very close. And now you know that the space shuttle uses rocket fuel, and that rocket fuel is hydrogen and oxygen. That's what's in the tanks that is used to drive the space shuttle up into space. So the potential energy stored in hydrogen and oxygen is very close to the potential energy that's stored in NADH and oxygen. So if we allow NADH to react ultimately and form oxygen, we get a release of free energy that's big. So 53 kilocalories per mole is a lot. All right. Now, let's look at what we actually do in order to do something. So, you've had your lunch, and what's mainly in your lunch is macromolecules, starch, and proteins, and fats. And you break down those macromolecules to their individual subunits. These are downhill reactions, catalyzed by enzymes. They happen outside of the cytoplasm, typically outside of your cells, typically in your digestive tract, mainly. Because what you take up in your small intestine is the subunits, the amino acids, fatty acids maybe. I'm not sure how you take up fats, honestly. And the sugars, right? So you take that up. Now, if you're thinking about a cell that is digesting another cell, like a macrophage that digests a yeast cell, it will actually digest that cell, that other cell, in its lysosome, which is still outside of the cytosol. So the digestive enzymes are in the lysosome, still outside of the cytosol. So, stage 1, breakdown of macromolecules outside of the cytosol. Makes sense. You don't want digestive enzymes in your cytosol because they break down your macromolecules, which you value. Okay, then the second stage is the breakdown of those subunits. It depends on which subunits we're dealing with. In this class, we're mainly going to look at the carbohydrates, especially at glucose. Which is broken down in stages, ultimately forming acetate. Then acetate is bound to a coenzyme called coenzyme A. Acetate is the smallest fatty acid you can think of. Very central molecule in metabolism, acetate. So stage 2, breakdown of subunits to acetate. And in a final stage, that acetate, typically in the mitochondrion, is broken down. In that process, CO2 is formed. The hydrogen on that acetate is captured in NADH, coenzyme. That then is used to feed an electron transport chain, where ultimately that hydrogen is allowed to react with, should say that differently. Ultimately, the electrons from the hydrogen are fed into an electron transport chain, where the electrons are allowed to react with oxygen in the end. At the same time, a proton gradient is built up. Proton gradient, that means we're transporting hydrogen ions across a membrane, the mitochondrial inner membrane, that generates a proton concentration gradient and a membrane potential. Together, a proton motive force. And that proton motive force ultimately drives the formation of ATP. So that process, in the end, is called oxidative phosphorylation because we have to use oxygen to accept electrons from the electron transport chain, which allows the formation of the proton gradient, which is needed for the addition of phosphate to ADP to form ATP. The addition of a phosphate to ADP, that's phosphorylation.